Hello everyone. Today in this video we're going to discuss about one of the most important biological molecules, and it's called the deoxyribonucleic acid. Now, every biological molecule has its subunits or has its basic substance out of which it is made up of. And same things goes for the deoxyribonucleic acid, because deoxyribonucleic acid is a very large and complex kind of molecule, and it also has its subunits, or the word which I wanted to introduce you, it has its basic, let me pick a color, it has its basic subunits or monomers out of which it is made up of. Or monomers are the basic subunits of any molecule, and the monomers of DNA is called a nucleotide. It's called a nucleotide. Now, and nucleotide, if you observe a molecule of a nucleotide, which I have a picture right here, a nucleotide is basically made up of three basic parts. It has three basic parts into it. Now, the first part is the phosphate group. The first part is the phosphate group right here. This one. Now, it's a derivative, or it's derived from a molecule which is called the phosphoric acid, or H3PO4. So here it was a hydrogen, here it was a hydrogen, here it was a hydrogen right there. There were three hydrogens, and this phosphate group is a derivative of this phosphoric acid right here. So this thing right here has acidic properties, or the word acid comes from this phosphate group. The second part is the sugar molecule. Now, it's a ribose sugar, which means it has 5 carbons in it. And so if you want to count the carbons right here, so the carbons would be, so each end represents a carbon. So this is a carbon, this is a carbon, this is a carbon, and that one right here is a carbon. So how many carbons do we have? 1, 2, 3, 4, 5. So we have 5 carbons. So it's a ribose sugar. So here is the word ribo in the name of the molecule. So if you notice, it says deoxy, which means one oxygen is missing, and the oxygen which is missing should be right over here. So this oxygen would be missing, so then it would be called a ribonucleic acid, which is another molecule actually. So this one is called deoxy because it's lacking one oxygen. And the third part of the molecule, so this ribose, this goes from this is coming from here. And the third part, which is called the nitrogenous base, and basically this is one of the most common knowledge which anybody, if you're dealing with DNA or if you're studying genetics or any kind of biological branch which is related to heredity material, this is the most common knowledge everybody has. So nitrogenous bases, there are basically 4 kinds of nitrogenous bases. One of them is called adenine, other one is called guanine, and the third one is called thymine, and the fourth one is called cytosine. These 4 are the nitrogenous bases, these 4 are basically the nitrogenous bases, and so these have specific pairings with each other. So they pair with each other in specific forms, such as adenine pairs with thymine and guanine pairs with cytosine. So each nucleotide, so there are basically 4 kinds of nucleotide which can combine together in a lot of ways and a lot of combinations to form different kinds of proteins and different kinds of proteins which encodes different kinds of information. Now, if I have a picture I copied this picture from Wikipedia, and this shows the 4 nitrogenous bases. So this right here is a nucleotide. This right here is a nucleotide, and then this one right here is another nucleotide. So this one here is another nucleotide. Now, as I said, adenine pairs with thymine, guanine pairs with cytosine. So this is guanine and this is cytosine, and this is adenine. The green ones is adenine, and I think it's a magenta or purple. This one is a thymine. Now, now the thing which I wanted to bring into your attention is that these two molecules right here are basically, if you notice in the diagram right here, adenine and guanine, there is one kind of similarity between them, and the similarity is that these two right here are double ring. They have a double ring of carbon. One ring is over right here, and another one is here, and one is here, and another one is here. So these two, the double ring nitrogenous base pairs are called nitrogenous bases are called purines. And the ones with single ring, like this one and this one right here, are called, which is cytosine and thymine, which are called pyrimidines. Now, so these are the four kinds of base pairs which can form, which can combine together in different kinds of combinations to form different kinds of proteins, which helps in all kinds of functions within the cell. So very important molecule. Now, these so another thing which I wanted to bring into your attention is that this one right here, if you look at this phosphate group right here in this molecule, this one right here, let me pick another color. This molecule right here and this molecule. So this is the phosphate molecule, and this is the 5-carbon sugar molecule. So this is another phosphate molecule. This is another carbon sugar molecule. So it forms some. So you can see a pattern right here. There's a phosphate sugar, phosphate sugar. So it forms a backbone of the DNA molecule. Now, the structure which you are seeing, the DNA molecule, is not basically its structure. This is the unwound state of the molecule DNA. The molecule DNA is actually some sort of like a candy, when you a wrapped candy. So it's basically, its structure is something like that. So you can imagine that this structure which I am drawing here is like the this is the wound stage of the wound structure of the DNA, and this one is the unwound slice. Sort of, you twist it around to open it up, and each of these strands, or each of these nucleotides join together, like this strand right here with these whole things with the nitrogenous base, with the sugar and the phosphate. This whole strand is called a helix, and the DNA is a double helix molecule. Now, if you notice something in the structure right here, there is some sort of pattern, like if you notice, like if you give me just one strand of the DNA molecule, I can give you the other strand without even looking at it. And how does it work? Look, if like you see that this one is, the green one here is adenine, the red one here is cytosine, the magenta one or the purple one is thymine, and this blue one is guanine. So if you give me this strand, I can automatically say, okay, adenine pairs with thymine, cytosine pairs with guanine, and thymine pairs with adenine, and guanine pairs with cytosine. So these two strands run anti-parallel. They both are anti-parallel to each other. One is running in this direction, and the other one is running in the opposite direction, which is really useful when they are multiplying or copying themselves, and hopefully we'll discuss that in a later video. But in this video, so this was an introduction to the molecule of DNA. Now, the double helix structure, so let me write that down. The double helix structure of the DNA was given by double helix structure of the DNA was given by Watson and means Watson and Crick, which they won the Nobel Prize in 1962 for that. So these two personalities right here won Nobel Prize. Nobel Prize in 1962 for that. And when they gave this structure, in 1953, and in 1953 they also mention that, and this is their quote actually from one of their works. It says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So what do they mean by that? It means that when they gave this structure, they eventually came up with an idea. Oh, the way they gave the structure of the DNA, it's also helping them to suggest a possible copying mechanism for the genetic material. I mean, this thing helps to make a concept, or create a concept in our minds that, oh, how does this DNA generate? Obviously by observing it too. So what's the mechanism of the DNA, how does it replicate itself? So hope you found that these things useful, and which is one of the most common knowledge that you must have in order to study DNA. And just for a reminder, deoxyribonucleic acid is a polymer, or like this one's, if you call this monomer, the monomers join together to form polymers. So many monomers join to form polymers, and obviously DNA is a huge molecule, so it's a polymer. So now, so what were we discussing about? Yes, how does the DNA copy itself? When they said it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And when they mean by specific pairing, it means that adenine pairs only with thymine and guanine only pairs with cytosine. Guanine only pairs with cytosine. So there is actually a theory which is accepted now. It's called the semi-conservative theory, semi-conservative replication. And replication means copying itself. Replication. And what it says that, what it basically is that if you give me a strand of DNA, so that's A, G, C, T, and C. So, and the other one would be just the opposite. So adenine pairs with thymine, guanine pairs with cytosine, cytosine pairs with guanine, thymine pairs with adenine, cytosine pairs with guanine. So so let's just suppose that this is the original strand of DNA, which is not actually the strand. I just made this up. So it eventually cuts itself in half, and it doesn't do it by itself. Specific enzymes are responsible for that. So it cuts itself in half. So no, that's C. And just let me draw the other one right here. C, G, A, and G. Now, what semi-conservative replication means that when DNA, when the DNA molecule the strands are separated by each other, and these right here, when they're separated, means that these bonds, which are called hydrogen bonds, are broken off, and what happens is that another nucleotides come and attach themselves to that. So if the DNA is replicating, so these are two separate strands now. So the A becomes the T comes being attached with A, the G comes the G is attached with C, cytosine is attached with guanine, thymine is attached with adenine, and C is attached with G. And in this strand, adenine comes attaching itself with thymine, guanine attaches itself with cytosine, cytosine attaches itself with guanine, and thymine attaches it with adenine, and the last one, cytosine, attaches itself with guanine. So what semi-conservative means that the original strand of the DNA half of them remains the same, half of them is conserved, and the other one, the new strand, comes and joined together. So in this way, the half of the original parent of the DNA is remains the same, and in this way the DNA makes exact copy of itself. And where does this replication helps? Well, it helps in cell division when the cell is dividing, especially the what kind of cell division? Well, it happens in mitosis. Mitosis, it's a kind of cell division, and meiosis. So replication is useful when cell is dividing. So this is an introduction of the DNA molecule. I hope you found that useful, and in the next video we're going to study about in detail how the DNA molecule replicates itself. So see you in the next video.